Welcome to the fascinating world of the genes with Rohini, and today we are going to learn about the no-go decay, which is one of the mechanism of the mRNA surveillance. In detail, we have seen about the nonsense mediated decay, non-stop mediated decay, and today we are going to learn about the no-go decay. All these three mechanisms, they are the translation coupled RNA quality control system. That is, it is going to check out the quality of the mRNA. If the mRNA it is of the good quality, or is it is going to give the functional product or not, it is going to be checked. And if it is not going to be so, that is the mRNA, when it is a defective one, then it is going to be cleared by one of these mechanism accordingly the condition of the mRNA. Now, today's video, no-go decay. No-go decay, it is the pathway which is allowing the degradation of the mRNA when the ribosome it has been stalled. Okay, so when there is stalling of the ribosome during the elongation, at that time the endoribonucleases it is going to cleave the RNA, and after release of the ribosomes, where after dissociation of the ribosomes, that mRNA it is going to be cleaved, it is going to be degraded. Okay, now how the no-go decay pathway it is initiated, or what triggers the no-go decay? In the East, it has been observed that when there are the stem loop structure, when there is the presence of the arginine or the lysine tracks, or when there are the long stretches of the adenine residues, when there is a presence of the premature termination codon, or there is a presence of the rare codons or the chemically modified mRNA, it is present at that time, the no-go decay pathway it is triggered. And here it is observed in the plants that long stretches of the A, it is going to trigger the no-go decay pathway. Now, how the no-go decay pathway it is going to work, that is the mechanism of the no-go decay. Here we are going to see the mechanism of the no-go decay in the East. Here, look at this diagram. What you observe in the diagram, here it is the mRNA, it has a stem loop structure, the ribosome it is present, that is a translation it is ongoing. This it is the elongation process in the translation. Okay, so here ribosomes they are translating the mRNA, and now when it is coming across this stem loop structure, what you observe, that this ribosome it has been stopped. Why it has been stopped here? Because there is a no free mRNA which can enter into the ribosome. So this stem loop structure it is halting the movement of this ribosome, and thus this ribosome it has been stopped here. So here when there is this stalling, when there is the stopping of this ribosome, at that time what you observe, the condition it is like the termination of the translation. In the termination of the translation, what you observe, that the A site it is empty. When you look at, okay, memorize the termination of the translation. In the termination of the translation, at the A site there is a stop codon, and when there is a stop codon, okay, that stop codon it cannot be read by the tRNA because there is a no tRNA for the stop codon. Okay, so the A site it is empty, and this A site it is this empty A site or the stop codons which are present at the A site, they are going to be recognized by the release factors. Release factors they are loaded. Then there is a hydrolysis of the GTP. This polypeptide it is released, and now the unit it is consisting of the ribosome with the uncharged tRNA, and then there is coming of the ribosome release factors, and that ribosome release factors now it is going to do the dissociation of the ribosomes, and the tRNA ribosomes and the mRNA it is getting free. Okay, now this a type of an structure, but here in this case during the elongation, such a type of an structure which is going to be formed at the termination, it is going to occur here. But here in this elongation, there is a no stop codon. There is a presence of the codon, but the A site it is a empty A site. Okay. Now, as the A site here, it is empty, it is going to be recognized. Okay, when there is a elongation, when there is a elongation of the translation, ribosome, it has been stopped. A site, it is empty, and this, it is a signal for the no-go decay. Means no-go decay pathway, it is going to work where? During the translational elongation, when the ribosomes, they are stalled, at that time no-go decay pathway, it is going to be triggered. And to release this translational machinery, okay, to release this translational machinery, to recycle this ribosomes, there is the use of this Dom34 and the Hbs1. What is used? The proteins which are involved into the no-go decay in the yeast, which are that? They are the Dom34, and the Hbs1. This Dom34 or the Hbs1, one of these, or there may be the another nucleases which is going to cleave upstream of this ribosome here. Okay, this, it is a ribosome, this, it is a RNA, and upstream of this there is a cleavage. Okay, and after that, okay, after that, dissociation factors, the ribosome dissociation factors, they are going to come. It is going to do the dissociation of this translational unit. The mRNA, it is getting free, ribosomes, it is getting free, the mRNA which are getting free. Now, as there is a cleavage in this mRNA, the RNA which are released, they are in the two fragments. Okay, and these two fragments of the mRNA, now it is going to be chewed by whom? By the XRN1, which is a 5' to 3' exonuclease. And here there is a exonucleases, it is a SKI7, which is going to cleave the mRNA in the 3' to 5' direction. In this way, such a RNA which has the stem loop structure, intermolecular stem loop structure form, or the intramolecular stem loop structures which are formed, that structures which are not allowing the which are not allowing the completion of the translation, that is, which are not allowing the translation to come up to this stop codon, such a type of the RNA, it has to be cleaved. Such type of the ribosomes which are engaged into this translation, it has to be released, and for that purpose, this no-go decay pathway, it is going to work. Okay, so this Dom34, okay, this Dom34, it is going to work like as an release factor. Okay, it is a homologue of the release factor, and in the plants and the mammals, okay, in the plants and the mammals, the homologue of the Dom34, it is the Pelota. It is known as the Pelota. And this Pelota, it is like whom? It is like this eukaryotic release factor. In plant, it is observed that this Pelota, okay, this Pelota, it is a product of the ty5 locus. Okay, so this Pelota, it is like the Dom34, and it is going to work into the no-go decay, where? In the plants and the animals. Now, what is the mechanism? How the Pelota, it is going to involved into the no-go decay pathway? Just now we have seen in the yeast, in the same way it is going to work. Okay, so here there is a stalling of the ribosome during the elongation. Okay, during elongation, when there is a stalling of the ribosome in the mammals and in the plants, at that time the proteins which are going to come here, it is the Pelota. Okay, Dom34, it is in the yeast, but the Pelota, it is in whom? Pelota, it is in the mammals and the plants. So the Pelota with the Hbs1, it is going to come. Pelota, it is like whom? Pelota, it is like whom? It is a eukaryotic release factor. So it is going to bound at the ribosome. Okay, so Pelota and Hbs1, it is going to bound at the MTA site. Okay, it is going to bound at the ribosomes. And like the termination, there will be the GTP hydrolysis. Okay, and after the GTP hydrolysis, Pelota with the help of ABCE1. ABCE1, it is whom? ABCE1, it is a ribosome dissociation factor. Okay, so the pelota, with the help of the ABCE1, it is going to do the dissociation of the ribosome. So when there is a dissociation of the ribosome, okay, what is going to happen? The RNA, it is going to be released here. Okay, before the ABCE1 it is going to come, according to the research paper, the pelota, okay, the pelota, it is a endoribonucleases. Okay, it is going to cleave the RNA in between. So when there is loading of the pelota Hbs1, okay, when there is a loading of the pelota Hbs1, at that time it is going to cleave the RNA upstream of the ribosome. And when there is a dissociation of the ribosome, these two RNA fragments, they are going to be released. So here, these two RNA fragments which are released after dissociation of the translational unit, that RNA fragments, they are going to be cleaved by the SKR7 in the 3' to 5' direction, and XRN1 in the 5' to 3' direction. Okay, so in this way, the RNA, it is going to be cleaved, the translational machinery, it is going to be dissociated, the ribosomes, they are dissociated, and now that ribosomes which are released, that ribosomes, they are used for the next translational processes. Okay, have I understood up to this? Now, role of pelota, just we have seen its role. It is in the no-go decay pathway to release the ribosomes, and here the role of the pelota, it is in the recycling of the ribosomes. How? Pelota with the help of whom? ABCE1. ABCE1, pelota with the ABCE1, it is allowing the dissociation of the ribosomes. Now here, role of pelota in begomovirus resistance. How the pelota, it is going to give the resistance for the begomovirus. Now, what is the begomovirus? Begomovirus, it is a Gemini virus, and here some of the species of the begomovirus, they are going to infect the tomato plants, and the yield of the tomatoes, it is going to be reduced. So there is a need of the varieties. There is a need of the varieties of the tomato, okay, which are resistant to the begomovirus. It is observed into the tomato that the locus ty1, ty2, ty3, ty4, and ty5, they are going to show the resistance for the begomovirus. Okay, in that ty5, okay, ty5 locus, it is coding for the pelota. Okay, what? ty5, who? ty5, it is encoding the pelota, and the recessive form of the ty5, recessive form of the ty5, it is involved into the resistance for the begomovirus. ty5 recessive form means when there is a defect into the ty5, it is showing the resistance for the begomovirus. Now the gene, it is a recessive, there is a mutations into the pelota gene, that is a ty5 locus. So there is what? There will be the lack of the pelota protein. Yes, there is a lack of the pelota protein. Now it has been hypothesized that how the pelota, it is involved into the begomovirus resistance. It is thought that, or the hypothesis it is that, when there is lower levels of the pelota, or there is no pelota, there is a lack of the pelota protein, at that time there is a no recycling of the stall ribosome. Just we have seen that what is the role of the pelota? Its role, it is in recycling of the ribosomes, and its role, it is in the no-go decay pathway. So the ribosomes which are engaged into, okay, the ribosomes which are engaged into the translation, that ribosomes, they are not recycled. Okay, the stalled ribosomes, during the stall ribosomes, that is whenever there is a long stretches of the A, the ribosomes, they are engaged into that translating that mRNA. That ribosomes, they are not recycled. There is a no recycling of the ribosomes at all. So no recycling of the ribosomes, there is a no free ribosomes, they are available. Lack of pelota impairs the recycling of the stall ribosomes, so reduces the availability of the free ribosomes. Now in the cell, there is a no presence of the free ribosomes. What do you think? What will happen into the cell? There will be the efficient translation? No, there will not be the efficient translation. Why? Because the ribosomes, they are not available for the translation. Where they are engaged into the translation. They are not recycled at all. So that your subunits of the ribosomes, they are not dissociated. And as they are not dissociated, as there is a no free ribosomes, the translation, it is affected. And here, according to the research papers, according to the hypothesis, it is said that as there is a no free ribosome, translation, it is not going to occur. And due to that, the begomoviruses, okay, the begomoviruses translation, it is going to be halted. And as there is no translation, okay, as there is a reduced efficiency of the translation, there is a no progenies, they are going to be formed for the begomovirus. In this way, the recessive form, the ty5, that is a lack of the pelota protein, it is going to show the resistance for the begomovirus. Why? Because pelota involved into the recycling of the ribosomes. As no pelota, there is a no recycling, there is a no free availability of the ribosomes, no free availability of the ribosomes, translation, it is affected, and thus the translation for the viruses, that is for the begomovirus, it is also going to be affected. And in this way, the pelota, it is involved into the begomovirus resistance. I think you may have cleared the idea about the no go decay pathway. Okay, the Dom34, Hbs1, then there is a pelota, and how the pelota, it is involved into the resistance for the begomoviruses. If you are like my video, please subscribe my channel and also share my videos with your friends, so the knowledge, it is going to spread. Thank you.